end to APP. It was just much earlier in the cascade. And so I was scientifically interested in what was going on. So it took 20 years for Susan and her team to step by step bring in arguments and started convincing people, because there's a lot of people who have a huge interest in APP and tau, and don't easily let somebody new come in, as you know. And so I was interested in these little stones coming in. Evidence was consistent with that hypothesis. The real interest for me when I got closer to and got more involved when I first became an advisor was for drug discovery it's essential to have a tool to test the hypothesis. And NBD14 was that tool. And the proof of concept in vivo you saw with these cognitive tests, and you'll see them more in detail in the paper, maybe you can comment on them later. This positive proof of concept in an animal model with NBD14 was again consistent with the whole hypothesis. And the data we have at that point indicates that we must use this tool to see if we can make a medicine out of this whole hypothesis. Now, you know, life changes when you start thinking like that. Yes, very difficult. But you have to do it. One, Susan mentioned it, Alzheimer's is probably one of the largest medical needs in our societies that are getting always older. And anyone who can make a contribution, a positive contribution, will have a huge impact on patients, but also on society. Not to say they can make all the money COVID too, but that's not the primary thing now. With NB14, we are putting together all the data that we need to go to the next step. And the next step, in this case, before you can go to man, this is an experimental drug, and that's a drug that's never been tested before. And based on the mechanisms also, it's not like an aspirin or an anti-inflammatory drug where there are many examples and we know more or less what to do easily to move it on. Here we're going into new territory. What we do know is that by now, because of all these other drugs, the methods to test effect on Alzheimer's disease are quite well known and useful. That essentially what happens, the cognitive functions in Alzheimer's patients deteriorate faster than they would in normal age people. And scales have been developed that where you can measure that. And what you try to do is to find drugs that can reduce the gap between the normal and the deteriorating one. So that part, I think, will not be the major problem, difficulty. But before you start to go in these kind of clinical studies, you have to prove safety. Because, we want to help patients and not create new problems. Then we have no idea where this will go. We know that we have in animals a dose of NBD14 that can show this cognitive effect, positive cognitive effect, at doses where you don't see any obvious negative side effects. But that's not enough to go to man. You have to do a formal study. And what you aim for is what we call a therapeutic window. That is, you have to have a dose of this drug that can do the effect on cognition, can reverse the deterioration of cognition, but is far away from the dose where you start to see adverse effects. And that is what we need to do now, next, and we're planning these things, and that was quickly on the slides there at the end. These will take time, because all Alzheimer dependent models depend on deterioration of cognitive functions, and they take time, weeks or months in people. But you have to do it, because if you don't do it, it's clear we'll never know the answer. We don't know if NBD14 will be the drug that will make all the way to the end. You know, at this stage of development, the probability of success usually is not that high for all the drugs. What I like about this is that this drug is very much designed specifically for this hypothesis. So that's certainly an advantage. So that is where we are now, and then of course as a drug discoverer I'm totally fascinated what the answer will be. And that is why I agreed to become an advisor, to also try to help on the board to bring in this drug discovery aspect, because they are becoming now extremely important after Susan has given us the basic science arguments that we needed to get to this point.
Okay, so that gives you the clinical perspective from two experts who work for Novartis and GSK on how we can now take this forward to clinical testing. What I'd like to do now, as you can see, is move to the second part of the science session, which is to introduce Sara Garcia-Rattez, who will chair a panel on university collaborations with three of our very good and close collaborators. But I should say a little bit about Sara first, because some of you may not know her. Sara actually jumped ship with me from the university now 11 years ago, Sarah, isn't it? It's 11 years, yeah. So she came originally from Barcelona, having done her PhD at the University of Barcelona on the very target that we suspected the T14 worked on, the sacred alpha-7 receptor. So she was really in our sweet spot in terms of expertise. And since then I've seen her evolve and grow from being someone in a white coat working on tissue culture through to, apart from 2 periods off maternity leave, coming back, and now really being a formative core member of Neuro-Bio. She knows that, I have to say to people, she knows that the bodies are buried of everything. Yeah. So if you want to know anything about Neuro-Bio, ask Sara. And she now doesn't work in the lab anymore because she's moved to the more managerial level of coordinating the collaborations and overseeing the in-house drug development of the variants, as well as working with our so-called CROs, that's the commission research projects that we do. So I'm going to now hand over to Sara, who will introduce our distinguished panel. And yes, hope you enjoy. Thank you, Sara.
Thank you, Susan. So while my panel sits down, as Susan said, I'm the operations director, Neuro-Bio, and I've seen everything since its creation here. And today I would like to really thank you all for coming and joining us in this very special occasion. Our collaborators are one of the most important parts of Neuro-Bio. Without them, we wouldn't be able to validate our work, and the world wouldn't believe everything we do, as well as if we didn't have them. And so what I would like to do today is to have a discussion in which they quickly will introduce themselves, and then I will kick off with some questions about how we work together, and if we have time we can have 1 or 2 questions from you. If not, we can discuss it after in the break. So I would like to start with, please, each of you, if you could introduce yourselves and tell us what you do.
Hi, I'm Stephen Cobb. I'm a professor of chemistry at Durham University, and I'm the director of the Biophysical Sciences Institute in Durham also. I'm predominantly a synthetic chemist, but my group does a lot of chemical biology, so we're very used to working with biologists and medics. Our interests are predominantly around peptides and peptide therapeutics, and that's how we ended up getting involved with Neuro-Bio.
Thank you, Stephen. Zoltan.
My name is Zoltan Warner. I am working here at the Department of Physiology, Anatomy and Genetics. I'm also preclinical medical tutor at St. John's College, Oxford. And my laboratory is looking at how the brain is developing. Initially, you just have a monolayer neural sheet, and it's generating millions of neurons. They have to migrate to the right place, they have to interconnect, and then they have to signal with one another, and then process the information from the sensory environment. So what Susan mentioned, I'm very interested in what are the roles of certain molecules in delivering the cells, how they interact, and in particular, I'm very interested in what happens when you silence certain neuronal populations during development. And this is how I started collaborating with Susan and Neuro-Bio.
Thank you, Zita.
And my name is Tobias Bast, and I'm a behavioral and cognitive neuroscientist. I study brain processes and how they generate behavior and cognition. And we mainly do this in rat models, where we can combine selective manipulations of specific brain sites and measurements from these brain sites with behavioral tests of distinct cognitive functions that we are interested in, such as memory and attention and so on. And we try to use this to understand how this works in the healthy brain. But we are also interested in what happens or what must happen for these things to go wrong, and what's going wrong in human neuropsychiatric disorders or neurological disorders. And we've also started recently working with people where we use similar behavioral tests to our lab models, to our neural models, and then which we hope will help us translating our findings from animal models to people. And I think we say later what we do with you. Yeah, is that correct?
So I think now that we all know what you do and who you are, could you tell us what appealed you to Neuro-Bio?
Well, for me it was a serendipitous interaction. So we were filing patents and we met Anton. And Anton, this was about 4 years ago, and Anton said that he was also working with a company that had an interest in peptides. So we came down, to meet Susan and yourself, and I think there were two things. One is, I think when you meet Susan, it's, as we've seen today, it's very enthusiastic about the science, and I think that's very infectious, and you get sort of taken along by that. And secondly, as an academic, we develop a lot of chemistry that within our academic labs, we sell in grants as processes and technology that could improve drug molecules. And this gave us an opportunity to actually take our academic methodology and actually apply it to a more translational problem, which is exactly what we're doing. So I think there was the real enthusiasm, and I do genuinely believe in the science. I think there's some really exciting science. So the people, but also the opportunity for us to apply what we're doing in an academic setting to a real translational problem.
Thank you, Steve. Thank you, Anton.
So I have known Susan for a long time. I use her BBC television series for my teaching. I teach second year neuroanatomy. I have on the practical, so I knew Susan for a long time. But it was really, I think, Peter Sommerly who started looking at the T14 expression and noticed that T14 was expressed in a very interesting pattern in the developing brains. And this is how Peter showed me the sections, and then we became very interested to follow this up. And both in human and mouse and in rat. And also, while we were looking at T14, and I started to understand a little bit more of the biology from Susan, then we linked up these projects to my ongoing projects when we silence or stimulate a particular cell population in the developing or adult brain. So the last 10-15 years I've been looking at ways of so it's almost like giving Botox to individual cells. So you remove the protein called SNAP25, and then the synaptic vesicles cannot dock and get released with the action potential. So we are using this genetic model to study chronic silencing of certain neurons. So, and I was, through these mechanisms, I was always interested in neurodegeneration, because these neurons eventually will die earlier, the ones we silence. So, and we also stimulate these cells, genetically express some channels, and then we inject some drugs, and then they get activated while this is called chemogenetics. And so we discussed it with Susan and said, look, if we change the activity, and T14 is indeed so fundamental for activity in the nervous system, then it has to change in these models. And sure enough, they had a big change. So I'm am now hooked on, and I would like to know the answer to some of these questions.
Thank you, but we'll carry on. We'll carry on discovering more.
So I started working with Neuro-Bio. I think it was through a link in Nottingham, a former colleague of yours, and Susan was looking for somebody to work with to provide proof of concept for the idea that T14 really causes these degenerative processes in vivo, in a living organism, initially in a rat. And what was required for this was some methodology to apply this toxin specifically into specific brain regions, and then to see what the animal is doing and what's happening in the brain. And we are doing this type of work, so we got in touch, we discussed the project, and it's an interesting hypothesis that is pursued by Neuro-Bio. So it's interesting, it's appealing theoretically, so if you don't have a hypothesis, science can't really progress, so there's a clear hypothesis, and if the hypothesis is correct, it would actually be great, because then we would have actually an angle on treating disorders that cause a lot of problem to many people. So that was very interesting, and then we started collaborating, trying to provide proof of concept initially for the idea, and then looking forward, I think developing this animal model will also allow to test treatments, to test other treatments beyond what Neuro-Bio is currently developing. So that's how we hope that might develop the line of flux that we're involved in with Neuro-Bio.
Thank you, and I think Tobias just said it a little bit, but in reality what got us together is the curiosity to understand what's really happening with our hypotheses. But I think what everybody would like to know really is, what do you think the best outcome of the collaboration is going to be?
So we're involved in a sort of backup series of molecules, I suppose, or a series of molecules that are basically derived from MBP14. So I got involved after some initial work had been done, where the MBP14 had been simplified into a much smaller linear peptide fragment, and the challenge was to try and improve that small peptide fragment, look at making it more stable, and also improving the biological activity of it. So we've been making molecules, we've been sending them down, we've been catching up on some of the biology that's been done here, and we have some really quite interesting, nice hits, I think, coming out, and we have been able to increase the biological activity. It's always a double-edged sword, because if you're trying to improve biological activity and the stability of a molecule, you might do something that improves the stability, but then takes a hit on the biology. So we're constantly having to play both sides of the fence. But for us, we've been able to take some of the technologies and interests we have in peptide mimetics and bring those into the project, which has been really great for us to do that. So I think what we're looking at is being able to get a molecule or two or three molecules that are suitable to take into the in vivo efficacy studies. And I think we've got some nice molecules. We're fortunate that we have a new thing going where we'll have a student in Durham making more compounds. And so hopefully sometime, maybe in the next 18 months or so, we'll actually have some compounds to take into the in vivo part. And I think that would be a good thing to finish, or a good place as a next sort of hurdle for us.
Thank you, Steven. Also, I think as a small company, I think we're privileged with this collaboration because we have a lot of backup compounds. So we know that, if one fails, we have a list of several that can go straight to the next round. Thank you, Steven. Zoltan.
You heard the big picture from Susan, but my particular interest is very concentrated on the developmental role of T14, and then when it is switching into this toxic mechanism. So what is driving us in this collaboration? We would like to publish high impact papers, so I'm still an academic, that's what's driving us, unlike many of you, but so we want to discover some fundamental biological mechanisms, and I do believe that T14 has perhaps some very interesting role during development. And the other benefits we have from this collaboration is, and we don't really talk about this too much, but actually my students admire Susan, and also she's a role model for the students. I don't know whether you noticed, whenever we have meetings, I not only bring the students who are involved in the project, but I bring others also just to get exposed to the industry, to see a positive example, and to stimulate the group. So we very much appreciate that.
Thank you, Zoltan. Tobias.
Yeah, we so in our work with Neuro-Bio, we have recently seen evidence, and Susan showed the data, that if you expose this part of the basal forebrain to this toxic peptide for an extended period in rats, then this leads to some neurochemical changes that are consistent with very early stages of Alzheimer's. At this stage, our behavioral measurements haven't revealed anything strikingly different with these animals, so there are not any obvious cognitive impairments, also consistent with these early stages of Alzheimer's. So yes, the disease lying dormant in people that would be 10, 20 years, as Susan explained, and in the rat, you can mimic this precursor stage potentially. And now the question is, if we expose the brain for longer, do we ultimately see these behavioral changes, the cognitive changes that are characteristic of an early symptomatic Alzheimer's? And that's something that we would hope to show. And ultimately then the optimal outcome would be that we then test this potential treatment, and we find that the process can be halted. Then this goes into people, and stuff isn't, the treatment goes into people, it doesn't have side effects, and it makes people healthier, so that would be great.
Thank you, Tobias. And that wouldn't just help Neuro-Bio, would help, everybody, all the companies that make drugs for Alzheimer's actually would start testing what's the real development of the disease, and then, we'll all fight together. We wouldn't make as much money, but it would be enjoyed by everybody else. And I think everybody, including Susan, is making sound like everybody's easy, and we've got all these data, and everything is super easy to obtain and get positive data. But obviously, there's always challenges. What do you think was the biggest challenge in our collaboration and the work that we did together?
I think there's the work that we've done, which I think, as I say, is balancing off the stability. So just in very basic terms, if you have a cyclic peptide, the peptide tends to be more stable, and the enzymes within the body can't chew it up so readily, which is why MBP14 is a good structure, if you like, in terms of a peptide drug. When you break that cycle open and you look at smaller fragments, the smaller fragments are actually more susceptible to degradation. So in the work that we do, it's been that trade-off between biological activity and stability. But I think, just as a general comment, as a peptide chemist, I think one of the exciting things about what is going on here is that you have a good route that Bachem have developed to make the peptide, because doing that on scale is not easy. It's challenging in the lab, and being able to scale up that chemistry. So that's a big hurdle in terms of the peptide chemistry to overcome. And I think also the fact that the molecule survives for an hour or two and has that kind of half-life, again, is a big hurdle, because as a peptide chemist, two of the challenges is being able to make a molecule that's cyclic and actually having it having a reasonable half-life. So I think in the big picture, you've overcome two of the major challenges, and that's really exciting. And And from the work we do, it's being able to get that trade-off between stability and biological activity. But as you mentioned, we've got quite a lot of promising compounds to take forward, and are excited to start making the kind of second generation and third generation of these with the student in October.
Thank you.
I think for us the challenge is to link up the developmental steps to the final stages of neurodegeneration. And I think it is in general in the field, these fields are not linked up at all. You are driven by very short periods. You either study just the aging or just the developing brain, but nobody is really following this up. And I think we should broaden the horizon and look at these mechanisms. So perhaps some of the genes which are involved in early development, they might have a very important role in neurodegeneration later on, and that's why T14 is so interesting for me. And we would like to link up these two fields.
Thank you.
So in the development of the life model, what has been difficult is to find at which level, at which concentration this toxic peptide actually needs to be in the brain in order to cause some visible changes, first in the brain and then potentially later in behavior. And so to find this out is quite difficult. One needs to get certain concentrations into the brain and a certain repeat this potentially. So we've had a couple of shots, first a one-off application which had limited effects, and then we thought maybe if one extends this by doing repeated application, and that seems to cause a change now. Then the question now is if this is to result in behavioral changes akin to Alzheimer's symptomatology, then we would need to extend the treatment even further. And the other problem is always that the funding timelines are very short. Things need to be done very quickly, which I personally don't like too much. And it's also difficult when you do this in vivo work, because you need to get things in place, and you need to have people around who can do it. So yeah, these have been the practical challenges that we faced.
Yeah, I think if we had all the time and all the money in the world, to do it, then it'd be everything more, easier. But obviously, we have to really think through and just go on the spot. And I think perhaps my final question, just so we have time for some questions from our guest. If there was only one thing that you hope, people in the audience today would remember about this conversation, what would it be?
I think it's obviously MVP14 is the kind of star molecule, but I suppose I'd like to make a little plug for some of the molecules that we're developing. It sounds a bit self-centered, but I think it's interesting that MVP14 is a particular class, and we're looking at a very different class of molecules. So there is molecules being developed in the pipeline that have similar properties and functions to MVP14, but are a very different class of molecules. And I think that's important because, as it was mentioned there in the last speaker, it's a challenge to get things through the drug discovery process. So I think having backups that are structurally different and will perform in vivo in a different way is a really good thing to have, and we're excited to be part of the project in developing those.
Thanks so much, Stephen.
My message is very simple. If you want to understand final stages of neurodegeneration, you have to understand development. Because some of these mechanisms start there. And of course we now understand that autism, schizophrenia, attention deficit hyperactivity disorder, epilepsy, these have all developmental origins. But what is not commonly known, that some of these neurodegenerative conditions, they might have developmental origins. Last year there was a paper where they looked at in Huntington's disease, which is usually not starting before 30, 40 years of age, at embryonic week, 13 weeks, they could see some changes in neurogenesis. So I think what will happen in the field is maybe the next 5, 10 years, the attention will turn to development. that's why we are involved in this project.
Again, I think that's also a message. We wouldn't be able to understand our Jekyll and Hyde theory without understanding, comparing the embryological and the adult involvement of T14.
And that's why we have to keep going with some of the projects where we alter the activity. Either we elevate or suppress the activity of certain cells and have a look at the T14.
Thank you, Sultan. And Tobias.
One thing that we might want to take away is I did mention that things are difficult and take time, and so if those of you who support this sort of research could do something to extend the funding timelines, that would be good. Yeah, if you just give people more time to do the research, I think people are keen enough. It's not to take it easy, but it just makes it potentially more efficient. Yeah, if one doesn't just need from in 5 months, 6 months periods.
Thank you so much. And well, thank you so much, panelists. I will give now word to the public. If anyone would like to ask a question. Yes, please.
Hi. Being a clinician, I know there are about 60 different forms of Alzheimer's. And what is interesting to me is in pain medicine where I'm at, we have a multiplicity of pathways leading to a chronic pain state. So we have to find a common constellation of symptoms, but when you try and tackle one pathway, the others seem to carry on. So one of the tests I would do when you find a new pathway, say, okay, what are the major causes of clinically predictable Alzheimer's? Not the rare genetic ones, but the clinically predictable. And the two obvious ones are people who've got diabetes and people who've got hypertension. We know they've got an increased risk. Has anybody had a chance to measure either in animal models or in clinically the levels of T14 protein? And the other one, of course, which is very topical, is after traumatic brain injury, getting chronic traumatic encephalopathy.
So we haven't tested, but definitely is one of our theories that a blow in the head could induce some kind of release of the T14, the same as I'm sure Susan agrees.
Yeah, so basically any form of insult or damage, if it's in the critical area, would then trigger that mechanism. And of course someone who's hypertensive might have a subclinical stroke. Yeah?
Yes.
And that then would trigger that series of events.
So, for example, you have streptozocin diabetes-induced rats. To me, that would be the next thing I would do in the animal model. Is simply look for T14 levels when those rats turn from ordinary rats into diabetic rats, because it's like 50% of the population.
What do you think, Tobias? Was that something that we could do with time and money?
Yeah, with time and money you can do anything. I haven't worked with a diabetic rat model, but I think that makes sense. These models are available. I've seen sort of used in the context of diabetic pain, and yeah, that could potentially be done.
And similarly with hypertensive rat models, spontaneous hypertensive rats. So, that to me is actually, I would suspect the T14 is involved, just the way I've looked at the papers. But it would be a very powerful selling tool to all your investors here, including me. So you can say this is actually a final common pathway for the development of an Alzheimer's state in the future.
We know that only 10% of Alzheimer's is familial Alzheimer's, so we account for every 90%, the rest of the cases, we hope that T14 is involved. Thank you. Anyone else have a question? Peter?
Just a question for Stephen. With the additional compounds that you're creating, who owns the patent over that?
That's Neuro-Bio that owns patents for that. And I think Anton probably, I think they are patented. Yeah.
Anton.
Where is Anton?
The linear peptides I was talking about, yes, they are patented and they are owned solely by Neuro-Bio, but I know that you're making derivatives and analogues. They are not yet fully tested or protected. When we think they're worth protecting, if they work, then they would again be owned by Neuro-Bio.
Yes, so what we normally do is we test them through like 3 systems that we have in-house, and if they work in that 3 systems, if they pass, and they're similar to MVP14, that is what we told with Anton, and we have the data to put in the pattern. Because we create quite a lot of the variants. John?
Can I ask Stephen, could you use artificial intelligence to look for the active sites on the site plating and then find simpler molecules that will have the same active sites?
So yes, the answer is simply yes. We have applied previously for Innovate UK funding to do some of this, and I can talk more about this in the break, but we have been looking at using in silico modelling to see if we can, because the thing is, I could sit down and write out literally thousands of peptide molecules that we can make, and we have enough hits now that we have some commonality. We have been trying to do some in silico modelling to try and do this in a more rational, sort of computer-aided way. And I can talk more about that in the break.
As a follow-up, if you could do that, will you be able to patent the active sites, as it were, rather than the molecule?
I think that's a question for Anton. Perhaps I'll pass that one over.
Yeah, you can discuss over the break. And one last question. There was someone, Anne-Marie.
So you talked about designing the molecules at the moment to kind of target the receptor and become more efficient. I'm sure there's also half-life involved in everything. But what about metabolites? Because if it turns back into T14, then it becomes active in the cell again. So how are you tackling that?
I think we first need to test if they protect. If they don't protect, then obviously we assess that it could be that there's a test to look for metabolites of compounds. If we, for example, with MVP14, we did that initially. We looked at what happened when you put in blood, when you put in, mainly blood, and then you look if in the blood it's broken down into a toxic molecule. If that's the case, then obviously we wouldn't use it. But that's how we've tested before. Thank you. Well, thank you so much. Please, a round of applause for our. So we met 3 of our very important collaborators in the UK, but we have some collaborators that work on the oncology project, as Susan already presented. They live far away, so they can't be here with us today, in Spain and in Australia. They Chenopad and the Cancer Research Institute, the Olivia Newton-John Institute. So I will show you 2 videos that they will explain what they do with how they collaborate with Neuro-Bio. So Chenopad, the Anna Portela, the CEO, and Rosa Bosch, the preclinical project manager. And Chenopad provides services with cancer drugs and studying metastases, and especially in colorectal cancer with us. And Professor Robin Anderson and Dr. Victor Rebus, both researchers at the Olivia Newton-John Institute. There's the newest cancer research in Australia, and they're developing also treatments for cancer and metastases, and they're working on breast cancer. Apologies for the first video. At the beginning it's a little bit strange, the sound and the accent of Anna, but it gets better.
Hello, I am Anna Portela, the CEO at Chenopad. Chenopad is a CEO that was incorporated in 2014, and we're working mainly in the development of oncologic drugs in the preclinical phases. In our services, Russian-derived shareholder. So these are models of the recapitulated human disease because a human tumor, either from cell line or from operation sample, is grown into a living mice. In those mice, we test the compounds and see its effect and also its toxicity. With Neuro-Bio, we started a collaboration based in colorectal cancer models. And let me explain you some of the numbers of colorectal cancer. Colorectal cancer is the second most common cancer in Europe, accounting for over 14% of all cancer diagnoses across the country. One European dies every 3 minutes from colorectal cancer. The 5-year survival rate ranges from 90% if the disease is diagnosed stage 1, localized disease, to 14% if the disease is diagnosed at stage 4, which is metastatic disease. The compound Neuro-Bio is developing aims to treat those at stage 4.
Hello, I'm Rosa Bosch. I'm the project manager in Sinovac, and I work directly with the Neuro-Bio project. We checked the toxicity of MVP14 compound, and the results were very promising. Also, we did some experiments to check the effectivity, the anti-tumor effect of the compound on a subcutaneous xenograft model of colorectal cancer. And again, the results were very encouraging.
Here at Sinovac, we were looking forward to continue working in our project and aiming to the results to have the compound reach the clinical stages as soon as possible. Very regards for the Sinovac team.
Now just see the Olivia Newton-John.
Hello, my name is Robert Anderson. I'm a professor in the School of Cancer Medicine at La Trobe University, but working at the Olivia Newton-John Cancer Research Institute in Melbourne. My research interests are in breast cancer, in particular in advanced breast cancer. In other words, breast cancer that's spread to other organs in the body. It's interesting to note that back in the 1960s, about 1 in 20 women developed breast cancer. Today, 1 in 7 women will develop breast cancer in their lifetime. So there's been an enormous increase in the incidence of the disease. But the good news is that we have much better treatments now, and we find that we can, at 5 years, we can have close to 100% survival for those patients who are diagnosed with early breast cancer. However, if the cancer is spread, which is a process we call metastasis, then the 5-year survival drops quite low, less than 30% over 5 years. So the real goal of my laboratory is to try and find better treatments for those people who develop metastatic breast cancer. So it's really wonderful that we can now team up with Susan Greenfield and her colleagues at Neuro-Bio and start testing a drug that they have found has good activity in Alzheimer's disease, at least in their preclinical models. And we do have some evidence that this same drug might have an effect in targeting cancers that have spread to other organs, in other words, cancers that have metastasized. And so we've just started testing this drug in our preclinical models of breast cancer metastasis. And so it's really great to think that we might be able to repurpose a drug that has efficacy in one type of disease that might also have good activity in cancer. So I'd like to introduce you to a senior postdoctoral fellow in my laboratory, Dr. Rick Redford, who's the one who's actually running the experiments using the Neuro-Bio compound.
Thank you, Robin. So we have a preclinical model of breast cancer that spreads spontaneously to distant organs, thereby recapitulating the human disease. So we have a molecular tag in our cancer cells that we inject that allows us to track the progression of the disease with live in vivo imaging. And it is our hope that by using Neuro-Bio's MVP14 therapy, we can interfere with the process of metastasis or the spread of the disease to distant organs.
Thanks, Rick, and thank you for this opportunity to let us tell you about our research in Melbourne, Australia. Thank you.
And now we have a break of 30 minutes, so please everybody be back at 3:30. And now you can go with the panel or with Susan.